All right, we're going to go ahead and get started. Welcome to class, everybody. My name is Karen Lowen. I'm teaching faculty here in the genetics department, and you are actually attending a class that I teach right now. So every spring I teach a class. It's a 1-credit seminar class called Confronting Eugenics. And in that course, we confront eugenics, and we start really in the 1700s, really. Talk about Linnaeus and everything. And we move through the history into the height of the eugenics movement in the United States, which was really in the early 1900s. But we go beyond that into what I call the era of the genome. And then we talk about modern genetic technologies through the lens of the history of eugenics. And in this course, I have a couple guest lectures that people I invite to come in and talk. And last year, and one of the people I invite every year is Casey Butcher, and she's fabulous, and she gives a talk on the history of eugenics at UW-Madison. And she's so good. She's so good that I invited her. I decided like more people needed to hear Casey than just my class. So I invited all the public and the whole department to come and hear her lecture last year. And it was well attended. And afterwards, our department chair, Francisco Pellegri, said to me, Karen, we need to have a seminar like this every year. And so was born the Scientific Accountability Seminar Series. And our department hopes to host a scientific accountability seminar every year. Maybe not always as part of my class, but this year it is part of my class. And so I had our speaker today is Dr. Matt Anderson. He's a faculty member here in the Laboratory of Genetics, and I had invited him to come and talk to my class. And after Francisco said, could we have a more formal seminar? I said, well, let's ask Matt if he'll just turn his casual talk in my class into something for everybody. And he graciously said yes. So let me tell you a little bit about Matt. And let me get my notes out here. So Matt was an undergrad here in genetics. So this for all of you undergrads here, you can see what can happen to you. So he graduated with a BS in genetics here at the University of Wisconsin-Madison, and then he went on to do a postdoc at Stanford. He did a couple, not a postdoc, he did his PhD at Stanford, and then he did a couple postdocs at the University of Minnesota and at Brown. And then he was hired to be a faculty member at Ohio State University. And somehow we stole him from them in 2023 when he joined our department. Research in his lab is focused on how genetic diversity contributes to variations in species populations. And he does this by studying the human fungal pathogen, Candida albicans. I don't think he's going to talk about that today. But there are also multiple efforts in his lab directed to empowering Indigenous communities in North America in engagement in research. And along those lines, he's a current affiliate member of the Carl R. Woese Institute for Genome Biology Center for Indigenous Science, which is part of the University of Illinois. So I think that is probably more related to what he's going to talk about today. And anyway, thank you so much, Matt, for being here today. And thank you all for coming.
Thank you. All right. Thank you, Karen, for the introduction. And I don't know, maybe this would be good if this happens to you, and maybe you want to do something else. We'll see what you all decided to go forward. So as was kind of mentioned, I don't work within human populations, generally speaking, but our work has moved into human populations. And I think a lot of this, what I'm going to talk about in my experience within the space, falls out of if you exist within a community that is often either explicitly or by circumstance targeted by genomics, you have to be thinking about these things within the context of your own community. And that's where my head kind of entered a lot of the thoughts that I'll walk you through today. So I had somebody from the custodial crew that I hadn't seen before. I think he like was roaming the halls after hours to see if he could find the person that was giving this talk. And he asked me if I was giving a talk about the genetics of vampires. And I looked into that last night, and there's a lot of information about that. So I do think like at some point we should have a genetics of vampires talk, but that's not what we're going to be talking about today. So what I'm really going to try and do is I'm going to try and provide a little bit of a primer about why there's so much interest in looking at genetic human diversity, kind of how that grew out of exploration of other genomic spaces, and then how there are a lot of interests working in that right now and thinking about kind of this last piece of the question, which is how does benefit get applied within those spaces? And how does that get distributed back out to the people that are being targeted or centered within these conversations? And so to start, it really comes down to the first whole genome sequence we had, which was this small microbial organism, Haemophilus influenza. It's got a small genome. It's only about 1.6 megabases, 1,600,000 base pairs. And this is done quite a while ago, right? It was in 1995, we had this first genome be published. And once we could do something like that, it became a question of, well, let's keep going. And those conversations actually started well ahead of the sequencing of this genome. But there's this really big energy that needs to be overcome to be able to move to something like humans. So here I'm just showing you in billions of base pairs the relative size of that Haemophilus influenza first genome, which again was published in 1995. And benchmarking these years is going to be important for thinking about where people's heads were at compared to humans. So a lot of questions started to get posed even before this about how do we move from something like this to something else, something that is bigger and potentially a lot more complicated. And what you ended up with is you ended up with this really kind of linear progression and working through genome sizes. So you had the E. coli genome come out a little bit after that. You then had the Saccharomyces genome come after that. And then you started having more multicellular organisms, mostly model systems coming out after that. And then finally that moved us into mammals, right? Including the human. And a lot of that tracks with the size of the genome, which honestly was a pretty fair predictor of the difficulty in assembling those kind of genomes. And so as this work was going along, to be able to get the human genome, they sought volunteers. So this is the advertisement from Roswell Park in New York, where they recruited people to be part of the first human genome project. It's important to note that there weren't a lot of stipulations to this, right? We need somebody that's an adult, and we need you to like not have undergone something that we know is inherently mutagenic, like chemotherapy. Outside of that, anyone could do this. And so there wasn't anything clear about like a compensation for this. It was just, we are interested in somebody that can perform this task of providing a blood sample. This was first kind of conceived of in 1988. So this is a fairly old approach. If you think about, again, when that first genome was published versus when this study was initiated, we're talking about a gap of 7 years. So this process moved across a lot of time. The amount of money that's gone into building the first human genome that started with this advertisement was around $3 billion, to the point that we had something that could serve as a solid draft. Most of the human reference genome that was built through the recruitment of those 20 individuals comes from a single person. 70% of the human genome is constructed from one. That person, of the 20 people that were recruited to this project, 19 were ethnically listed as white. One person was ethnically listed as mixed at the time. And so when you're looking at the draft human reference genome, 70% of that genome is coming from that one admixed individual. 30% is coming from the individuals that self-identified as white or Caucasian. So the genome was really interesting. What ended up being produced from that genome was a really interesting mix that provides genetic information, not of just necessarily one specific population, but potentially multiple populations, and subsequent analysis that you can find on the National Genome Research Institute's part of the NIH website is a schematic that kind of gives you a breakdown of within our understanding of like some of the contextual markers that delineate different populations. This is kind of the makeup of that human reference genome. This was a big deal. This is Bill Clinton announcing the completion of that first human genome in the year 2000. If you go to the NHGRI's website at this point for the 20th anniversary, they put out this massive webpage that lists all the different milestones, all the different people that were involved. It's interesting to think about this history. You see folks that are pretty familiar, like the longtime director of the NIH that's shown up there right next to Bill Clinton, right, as part of this public announcement. The Human Genome Research Project itself was started by, or at least the figurehead for this was James Watson. So from like Watson and Crick Watson. As time went on, he got backburnered in kind of these conversations for a lot of reasons that revolve around things that he says that are like incredibly problematic, which you don't want when you're trying to like build a human genome, right? But I think if we're thinking about that first advertisement for recruitment in 1988 to the year 2000, that's a 12-year process of collecting the DNA, assembling the DNA to a point that you have something that you think is solid enough to be able to put out into the public research space. It's important to note that there were two major updates to the genome for the human reference app through this. One was filling in the gaps that happened a couple of years later. And the second is the telomere to telomere build that I think was just published in 2020. And that was facilitated by using long read sequencing, not the, I don't want to call it short read, but the 1,000 base pair long sequences that were used to be able to assemble the human reference genome. A massive amount of money went into this project. I mentioned $3 billion. When the Human Genome Project was instigated, there was this idea of, well, we're going to do the science, but now we're not focusing on a bacterium or something that doesn't have like clear societal implications that every single person that encounters this is going to see. Now we're talking about humans. So a number of ethicists came along and said, we need to be thinking about how we're relaying and interpreting and contextualizing the information that we're going to collect as part of this project so that we can deliver it in a way that's not only like scientifically meaningful, but also able to be interpreted in a hopefully good way by the public, where they understand that this wasn't done in some way to take advantage of people, or that we're going to be dissecting people to then like parse out their information to set new insurance rates, for example, right? Which is a conversation that comes up a lot, and you will see it again in this talk. So the first time that an announcement was sent out by the NIH that was looking for somebody dealing with the LC components of scientific research, those are the ethical, legal, and social implications of science, was shortly after the foundation of this project in 1989. Okay, so a lot of that was put towards Stanford as kind of the hub. It's not Vivio Odawang. Mildred Cho became the first director of what was termed now the ELSI Hub, which is still an active part of the NIH's process at Stanford to oversee the ELSI components of the Human Genome Project as it was constructed. I think it's important to say really quickly that the ELSI side was always seen as an afterthought, okay? 5% of the budget was put towards the ELSI components, 95% was spent towards the sequencing. The recommendations of the first convening in 1989 around this were that it should be 50-50, okay? And obviously that did not happen. And I think it's useful to get a quote here that I pulled from this paper that's referenced down at the bottom that gives you an idea of the mentality of folks involved in the science on this project for the associated ELSI components that would come with publishing a human genome. So we have this report here. Andrews, who was one of the main people building out the ELSI program, reported that James Watson made a remark at a genetics policy meeting implying that he had configured the ELSI program to minimize its ability to impede the Human Genome Project, right? So I wanted a group that would talk and talk and never get anything done. And if they did do something, I wanted them to get it wrong. Okay, so that is the director of the Human Genome Project being quoted by the LC director being told, like, I don't want this, I don't care about this, I want it to be as irrelevant as possible. And that, I think, is a really important perspective because it gives an idea as we entered the genomics realm, the headspace that was coming out of some of the leaders there, even if they got backburnered. We see this happening across a lot of projects that were occurring simultaneously. So the Human Diversity Project was instigated around the same time, 1990, 1991. It was a global effort. Its goal, as stated, was basically to sequence the realms of human genetic diversity that exist before they got, quote unquote, lost. So there was this conception that different populations across the globe were going to become extinct. And by extinct, I mean intermixed with other populations, such that whatever unique genetic characteristics that may be there or may be thought to be there wouldn't be accessible to scientific researchers anymore to be able to explore. So that project actually was labeled where I got the title for this talk. It was called the Vampire Project, and I'll show you some of that a little bit later. The Genographic Project was meant to fundamentally do the exact same thing. It's really interesting just because of the timing around these things that we're going to talk about some of the flack the Human Diversity Project got, and yet you have the Genographic Project trying to do the exact same thing like 10 years later, right? And so while I might say within a genomic space, this is like ancient history, I want to make it clear that it's not, okay? The Helix program is the Genographic Project 2.0. Same leadership, same funding architecture, most of the same individuals involved in doing this, and they're continuing. Genographic Project got enough flack that they had to retire the name. Helix is what was born out of that ash as a really malformed phoenix, I guess. I don't have a good analogy for that. But I want to highlight a couple other projects that are working within the space of collecting lots and lots and lots of genomes that you can see represented on this campus, that we're going to talk about where I think there are some issues and some things for us to be thinking about. We're thinking about that benefit and how that benefit gets redistributed. One of those is the All of Us research program, which is funded by the National Institutes of Health. Its goal is around precision medicine, which we'll come back to. And the other one that most people actually don't think as much about is the Million Veterans Project. I was in the Marine Corps for 6 years as an infantryman. My sister works at the VA and is also a vet. So like, this one's actually been more successful because you have a bunch of elderly people that go into a clinic where you get free health care and you sign forms, right? So you just walk in, you're like, ugh, and off they go. And what they don't necessarily realize in a lot of instances is that they're signing up into this massive genetic study. All of Us was supposed to enroll 1 million people. It's not there yet. MVP was supposed to also enroll 1 million people. It's past the mark and it's now going to 2 million because it's at roughly 1.3, 1.4 million enrolled individuals to date. So this continuance of attempts to be able to access diverse genomes and use those genomes for different things is not past history, it's current. And so why does this become a bit of a sticky situation for folks to find themselves in? I'm giving you the leader, Dr. Cavalli Sforza's exact words that revolve around his context of the Human Genome Diversity Project that was first initiated. We have populations coming from across the globe. What you'll notice is that most of them are clustered in the global south, right? So this is, again, a question of who is this project for? Is it for us, by us, or is it on you, for me? Okay. I'm not going to read that. You can read it. Okay. But again, it's this idea of we need to capture these distinct populations before they go extinct, which again, does not sit very well with a lot of folks. And so there were a lot of very strongly worded responses to that. And so you have one from the Central Australian Aboriginal Congress, which is shown on top. This is where Vampire Project got its label. The Human Genome Diversity Vampire, quote unquote, project is legalized theft. That is a verbatim quote from their statement in opposition to this program. You see one from the Declaration of Indigenous People of the Western Hemisphere. Regarding that, we demand an immediate moratorium on collections and or patenting of genetic materials from Indigenous persons and communities or individual researchers. So they said this in part due to responses to 2 attempted patents from Indigenous people based on gene patenting that was becoming quite a rage in the early, mid, in the late 90s. There is legislation in place that basically makes it clear that this is not allowable anymore, but at the time that did not exist. And so there were cell lines and genes that were being attempted to be identified for different HTLV. These are different viruses that endemically infect human populations. From a Guayama woman, and they're down in Panama, and a Hagai man that is in Africa. So there was attempts to be able to profit off of collected materials from Indigenous folks already. And their response was, don't do that, okay? Enough public pressure was mounted that this is the origin of, in part, the UN's Council on Indigenous Peoples, as well as the UN Declaration of Indigenous Peoples' Rights on GRIP. I screwed that up. I transposed something in there. These are the origins. It was actually genomics that pushed the UN to build an Indigenous Peoples Council because of how much this stirred up folks across the globe. I think it's important to say that this is not a statement that any community is in opposition to scientific research, right? It becomes a question of how that research is done. And I just showed you the Central Australian Aboriginal Congress's statement about it being Vampire Project. This is also in that document. They're not saying don't do research. They're saying let's talk about how it's done so that we're not just getting extraction and we're getting something out of our participation that you're very interested with us. These also led to things like the Nagoya Protocol in 2010. And the Nagoya Protocol requires or sets an expectation of benefit sharing to populations that take part in this. This is an expansion of the Convention on Biological Diversity that was made by the UN about 12 years before this because of extraction of non-human materials from Indigenous communities. The most famous case of this is, and I should have had this in my notes, the San people in, I think it's South Africa. There's a drug that we all use that was derived from traditional knowledge they had around using a certain tree in their community to resolve symptoms. And I can't remember the drug, but if I said it, you all would definitely know it. So the Nagoya Protocol expanded that idea out to now people. As I mentioned, this really hasn't kind of gone away. I want to highlight really quickly some attempts to continue this within human spaces that are going to be fairly familiar. And then we'll transition a little bit out of what is mostly public sector and a private sector. And so the goal of the All of Us research program is like, good, right? Like, these are not bad things for the most part. The genetic ancestry, I would argue, like, we don't need that. But I also don't think we need to know why you love or hate cilantro. You could like cilantro, and that's great. You can hate cilantro, and that's okay too, right? Anybody can have whatever opinion they want on cilantro. But high risk for certain health conditions, I don't think you're going to find a lot of people that are opposed to that. And how your body might react to certain medications, I don't think anyone's going to be strongly opposed to that, right? These are good goals. The All of Us research program published a paper last year that basically gave a benchmark on where their study is at. I went back and looked at the website a little bit yesterday to see how this had changed. So the number of participants has grown quite a bit since this paper was first published. So there's roughly, I think it's 800,000 now enrolled participants. They put forward a major push. My critique around the All of Us research program in part stems from before it was even established with funding awards. In 2017, I was sitting at the University of Arizona as part of a workshop that I currently co-direct called the Summer Internship for Indigenous People in Genomics. We had somebody from the University of Arizona that was actually the lead writer for the All of Us application that they had submitting an application for targeted recruitment of Native Americans and Alaska Natives within the United States. They had not talked to a single tribal community before they said, don't worry, NIH, we will guarantee you we will get American Indian Alaska Native individuals for your study that you want. So again, the foresight to be thinking about how you do this work is critical. And one of my critiques of the All of Us program is that didn't exist. And I don't think Indigenous folks are the only population. I'll wrap back to that as I get farther in. So the goal of precision medicine, which the All of Us research program is directed towards, is to be able to match your genetic markers to the appropriate drugs and the appropriate treatments and the appropriate indicators of conditions. This is a fantastic thing to do. So you can treat the disease, you can find the disease more, you can treat it better, and you can find conditions we don't understand. So instead of basically going into the doctor being told, you got a condition, we're gonna try some things, right? We're probably gonna like, it might work for you, it might not work for you. My mom has epilepsy. They tried, I don't know how many treatments, brain surgeries, vagal nerve stimulators, different drugs. I was born on a teratogen. That was kind of fun. So teratogens cause like birth defects. So like my feet are all weird. I'm not going to show you my feet, don't worry. But like they try things and see what works and what doesn't. It works sometimes for you and it doesn't a lot of the times. So the goal here is like we shoot and we hit, right? Fantastic goal. So now we talk about warfarin, right? Warfarin really put a lot of the scientific endeavors of the University of Wisconsin on the map because it provided a massive influx of funds for the university to really build out this strong research program. And a lot of that is why I'm standing here in front of you today. Wisconsin was the only college I applied for, and luckily the only one I got into, barely. That's a whole nother story. So warfarin stands for the Wisconsin Alumni Research Foundation, WARF plus coumarin, because it's a molecularly similar to coumarin. So you can actually take coumarin and you can translate it into warfarin through a set of chemical reactions. So identified through sweet clover disease in cows at UW-Madison. Sweet clover disease was already known before, but why it was happening was understood here. It's actually a combination of like decay and sweet clover. If you feed sweet clover to cows, they're going to be fine. But if it decays, now you got a big problem. They synthesized it as a chemical anticoagulant in 1940. It was used as a rat poison for a really long time. And then they started testing it in larger animals, and they found that larger animals actually do a really good job, generally speaking, of tolerating this. So it became like the most common blood thinner, and is still the most common blood thinner used across the world. I looked up the statistics on like how many doses are given, and it's like 1 billion, I think, is the estimate currently, doses per year. Warfarin's a fantastic drug that's helped a lot. So in the role of precision medicine, why am I bringing up warfarin? Because not everybody tolerates it the same. There are 2 genes that are really important for metabolizing this. Cytochrome P450, which is the CYP gene over on the right side, and the target of the drug itself, which is the VCORC1. And what this is showing is, depending on what your genotype is at a specific position within these genes, you need different doses of warfarin to correctly thin your blood so you don't get blood clots. So if you have a stroke, you want to thin the blood, make sure that you don't get secondary clots and you break things up. How do you treat the individual correctly? And that's really based on genotype. We didn't know that for quite a while. And so what you ended up having is people, especially Alaskan natives, have an overabundance of certain alleles, specifically that require lower dosing. And so when warfarin was being introduced into the Alaskan medical system and the Indian Health Service, they were killing dozens of people because they were overdosing with warfarin over and over and over. And that is kind of shown here. So you really need a low dose for Alaskan natives, and this is why precision medicine is absolutely critical. I want to say that there's been a lot of work on All of Us research recently. The number of publications that have been put out under the auspice of this program have more than doubled in the last 12 months, which is actually pretty incredible. We're now starting to get out some real interesting and important precision medicine pieces to this. So I think it's beginning to serve the purpose, and you can see that based on LDL. So, frick, man, this is what happens when you talk across too many spaces. It's like cholesterol, right? LDL, low. Thank you. So they could reconstitute the loci that are important for thinking about LDL levels based on genetic background, right? Fantastic. This is a great use of all of us. Does anyone remember what the first research paper of all of us was about? Any guesses? Ancestry, right? If the stated goal of all of us, which part of it is genetic inheritance, but is really trying to help people's health, the first paper, this Nature paper in 2024, contextualized a lot of it around ancestry, right? And it broke up basically different backgrounds and ethnicities, and basically showed through the visualization of their data this idea that people of different races and ethnicities are distinct, right? And you can probably see how that becomes a problem. And a lot of researchers did too. And I think this is useful to know. When something bad happens, there are researchers that are absolutely saying, wait. So this is a statement from Michael Eisen, who's a really well-known scientist and is very willing to speak out on a lot of issues, noting that this paper is going to set a precedence for different ethnic-centric and racist groups for the future, because this is scientific support for distinguishment of races, right? So we need to be careful. We need to be thinking about what we're doing. It's important to note that stuff like this was written into manifestos set by people that have taken action within these spaces as well. So it's not just could, it's a does. All of this work starts with consent. What does an individual allow and what does an individual not allow within work with their genetic materials? This goes all the way back to that original advertisement. We need blood samples from you to be able to do this thing. Can we do the thing? There's been a process in which there are different levels of consent that have been built out. Some of them are very case specific. We're going to do exactly this work and that's it. Some are more like, we're going to work in this topic, cardiovascular disease, and that's it. Some are going to be more broad, biomedical research, or like, we can do whatever we want with your material. So this idea of broad consent. We're going to talk about broad consent a little bit more. As you move down the scale, you lose the autonomy, and obviously you increase the specificity as you move up, right? So there's a relationship between you being able to decide how your information gets used that runs opposite to exactly what can be done. And we've seen a wave. I was going to use something different, but like this is a cool image. And there's been a fluctuation in the way consent forms have generally worked. At the beginning, a lot of it was very specific consent language, but it gets onerous. It's tedious. I have like 4 or 5 IRBs currently, and every time we do something new, I have to put in a new IRB because I have to rewrite the language and have to go back out and reconsent folks, and it's a lot of freaking work. And so you had movement away from that specific IRB where it became broad. We're not going to do that anymore. It's too much time. It's too much energy. We got to find these people all over again. When we consent them, we're going to say we can do whatever we want. Because of some of the things I'm going to talk about, we're starting to move back to a more specific consent. Okay, so I do think these things are shifting. And I'm going to illustrate what I mean by broad consent by showing you a couple examples. These are real examples from consent forms and materials that I've been involved in reviewing for companies. I've highlighted something here. You will have the choice to provide 1 extra blood sample to look at your DNA if you consent. You'll notice if you look at the rest of this form is not about DNA, it's about vaccine. So it's about response, right? You don't need DNA for that. But they're saying, hey, while we're at it, can we get your DNA too? You'll notice that this is a very small section of even what are we going to be doing with your sample. And then what is that sample going to be used for? If you go into the supplemental files that you can be given on top of this, so this is the consent form's about 20 pages. The supplement is about another 15 pages. If you go back into that supplement, you can get a better idea. Your samples will remain in indefinite storage for 15 years as defined by local regulations. Your samples could be used for researchers on COVID-19 and respiratory viral disease vaccine. So this study we're saying explicitly is in this area, but we're asking for some more DNA. If you want to dig into it, we're going to be able to work in all these other areas that like maybe you're not really thinking about now. And we can do this for 15 years. So we don't even know what we're going to do with it yet. And that's broad. Here's another one. Blood samples may be stored frozen by the company or any companies working for them for up to 25 years. We can use them to develop new vaccines or new and improved tests related to coronavirus or other diseases. Any disease. So you're going in for something that clearly is linked to viral infections and vaccination, and who knows what you're providing your materials for. This is part of another one of those consent forms. Once your health data has been shared with authorized users, it may no longer be protected by federal privacy laws. So not only do we not know exactly what we're going to do with your information, but we're not even going to tell you that we can guarantee safeguard of your information. So we're baking it in so we can't be held accountable. Broad consent. It's broad. So why would you pursue broad consent? There's a lot of money to be made. By having DNA information that can be used for whatever purposes, the estimated profits within gene therapy market spaces over the next, like 8 years, it's going up a lot. So a lot of this comes back to money. And there's not a lot of clarity as to exactly how this works. We're going to collect DNA, we're going to do stuff, and we're going to make money. We don't know what we're doing in 25 years. We don't know what we're doing in 15 years, but we know what we can do will make money. And if you look at vulnerable populations, again, a lot of my context for this is centered in Indigenous people, but I'm trying to present this in a way that doesn't explicitly say this is only for Indigenous people, because it's not. But you see some of this, the early consequences coming out with Indigenous communities because they were very vulnerable within these spaces. So you have cases like the Yanomami, who provided DNA material back in like the early aughts, and are sitting here 10, 15 years later being like, you all said you come back with information to help us, and we got nothing. And this is true across a lot of Indigenous groups across not only the globe, but actually the US. It's gotten so bad that the National Congress of American Indians, which is a somewhat informal governing body that convenes all federally recognized tribes together in the United States, put out this notice in 2019 to the All of Us research program that's fundamentally a cease and desist letter, saying you're attempting to collect information from representatives of our tribal nations as domestic dependent nations. We have governance that you can't touch, and we're telling you stop. Okay. So you haven't talked to us. We don't know where the data is going. Don't do this. But it doesn't require these large research or federal programs. Folks are giving their DNA away. So if you look at the DNA market, it's grown substantially over time. I haven't been able to find any projections that move past 2019. That's when a lot of this data was collected, looking at these trends. But even in 2019, through a number of these companies that still exist, 50-fold increase in available genomes occurred over those 6 years. They're existing in a private sector, not a public sector, right? So those are explicitly going to be monetized. 1 in 5 citizens had taken a direct-to-consumer genetic test in 2019. And that one person is often contributing to many, 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 many studies. And you see this continuing into the present day, despite some of the different things that are going on. This is an advertisement from Biocertica that's like, for the holidays, what could be better than exploiting your family? Which, like, I mean, I get it's great to walk into a relative's house and be like, I don't have to cook, all the food's already there. But that's a little bit more of an understanding that it is this. I mentioned the importance around gene therapy and how that information can be monetized. I think it's important to say with direct-to-consumer testing, this is not different. 13% predicted annual growth within these spaces globally for the next 5 or 10 years. It's a massive market of over $1 billion in 2019 when they took a really hard look around this. If you look at specific examples, we have examples of DNA testing companies getting bought. We have GlaxoSmithKline buying a smaller company for $300 million around 2016. And we have Blackstone, which is the largest pharmaceutical corporation in the world, that bought Ancestry.com in, I think it was 2019, for $4.7 billion. So the information that you're getting from your DNA test for fun is being used in ways to get sold back to you. I'm not going to spend too much time on this. It talks about what your DNA could be used. These are terms from Ancestry.com. You can go look this up on their website. I did. It makes it clear that your DNA could be used for all kinds of different stuff, right? Beyond what you think you're using it for. And that you will receive nothing from any of the work that we do with your DNA. You paid us to do this, and now we're going to make money off of you paying us to do this, right? And I don't think that people necessarily always understand that. So your information is going to be collected. It could be used in all kinds of work. And we don't know what that looks like again. And we're not going to really make it clear to you that it's being used in that work. So it could be used for research, drug design, any commercial activity that we decide. And there are red flags specifically within this language that other people have kind of noticed. One here is Ancestry will endeavor to provide consistent quality and improve its features and services. Ancestry may use my DNA data to create new product features and services, including products related to wellness and health. You have to consent to be able to use the service. Okay, so you have, the only way you can do this is by opting in. So where does that leave it? Leaves us with 23andMe filing bankruptcy a couple weeks ago. So what happens to your information after a company that stores what is a massive amount of data like this goes under? And those questions are currently being raised a lot. And so you have a lot of articles such as this one that's basically saying like, well, we've seen what's happened in the past. You may not realize what's happening now. 23andMe is already contracted with GlaxoSmithKline to provide your data before it went under. This has been going on for years. So all this urgency around articles saying like, go in and remove your data and deconsent, it's too late. Doesn't matter. They already got your data. Okay? If you're not reading the consent, they can choose who to partner with, and they can choose what happens to the information as soon as it goes out. Okay, so I told you gloom and doom for like 30 minutes. I don't like using AI because like it's not good for the environment, but I couldn't find vampire eating money images. And so like I had to generate one. So I think the question is, where do we go from here? I don't want to leave us on the sour note of like, everything is doomed. Nothing is actually providing community benefit back to the people that participate, because that's actually not true. So where can things be done in a way that more equitably mirrors the input and the process in which people participate in these studies? And I want to highlight a couple examples. I'm going to start with industry. There are companies that are doing this in a better way. I want to highlight one called Variant Bio. What they do is they partner with communities to be able to get genetic information to target things of interest to the community, but they do it with 2 additional hitches. They're going to make money off of it. They're a for-profit company. 3% of all profits go back to the community that participated. Benefit sharing. It's not that hard. It's 3%, right? And 2, any products that they produce will be given out to the community for free. Variant Bio has been around for over 10 years. This model can work. You could set up autonomous research centers. Communities themselves can build and invest in areas that they want their work to do. I'm going to highlight one here called the Native Biodata Consortium. I guess as a disclaimer, this feels weird. I am on their board. That's not me saying like you should work with them. You can. The director is actually over across the street in the WID. He's giving a keynote at the Indigenous Research Forum. He's fantastic. If you decide you want to go over and hear more about that, it's right across the WID in that rotunda room in the center of the building for the rest of the day. You can talk to him about how he set up an autonomous research facility within the community that he lives in to address health concerns in that community. And this can be replicated in other contexts. I've been speaking about Indigenous people. I'm telling you now that there are Black and African Canadians that are setting up a very similar system that don't have this nation-to-nation relationship status in Quebec. So different communities are taking advantage of these systems to build their own infrastructure to target things that they care about. And the last is think about supporting health organizations. Organizations that contribute a lot of their funding specifically to addressing health needs that exist within the community that they're centered for. Most of these communities, American Heart Association, American Cancer Society, Epilepsy Society, you can track where their spending goes, and on average, around 80% of the funding goes directly back into supporting research, education, and training around the issues that affect those groups. It's a great use of funds. Okay. So I think that's all I need to tell you. I'm close to about where I hoped to be at. I think it's important for me to say, as you heard, I study fungus, and that's great. But I've been pulled into these spaces by a lot of friends and colleagues here because it affects communities that I'm accountable to and communities that I care about. And I think it's useful information for other folks to know so you can make those decisions for yourself, right? Maybe that you're fine with that. That's great. That's okay. But everybody should be able to make that informed decision for themselves. Thanks. Are there any questions? Yeah, Tam.
Mm-hmm.
Yeah, I think, yeah. So there's the conversation today around human information was covered in the Nagoya Protocol, which was born out of biodiversity, right? And that you're seeing the same thing here within human materials. Those conversations have existed in non-human systems for a long time. And I kind of like put my hands up because I had a conversation Monday afternoon with a consulting firm for the Microbiota Vault, which is trying to store and bank microbiome samples from global groups for like the preservation of mankind. So it's taking in specifically the papers, especially the early work for the Microbiota Vault made it explicitly clear they're taking indigenous samples to help urbanized populations, so industrialized groups. So I think these same ideas can work in a lot of spaces. It's just a matter of, yeah. it's all about probabilities and frequencies, right? So I think it's fair to say different populations have different histories, and therefore that's going to be reflected in genetic markers. Valid statement. I think that means that when working with a certain population, you're going to have to be cognizant of those things. But making an a priori statement that because you're X, therefore Y, that's where you like clearly step into territory that's going to become a problem. Hopefully that helps. Yeah, people that do human genetics are going to be able to speak to this better than I can, but I think there are some good people in this department that will have a better sense for that. And I think a lot of focus from like Donna Werling's lab and kind of these contexts, more rare disease focused area, and Justin Walter, that makes a lot of sense to me as a good place to start. When you're dealing with highly complex phenotypes, it's going to be presumably a lot of loci that are contributing to it, and it's going to be harder to find something that meets precision medicine expectations. But again, this is not my space, so that is my superficial answer. Yeah, they don't exist. So those requirements don't exist. They don't exist in the US and they don't exist at the global stage. We had a speaker here in genetics last year that is targeting populations from the global south and doing it in a way that I would argue is not good. So they're finding people walking on the street and they're offering them 10 or 20 bucks for like a small blood draw, right? This is not a clinic. This is not truly informed consent because they're not working with an English-speaking population. And so if that is happening 12 months ago with the speaker here, there are no regulations. It's shaming. Shaming is the only thing that you have at your disposal right now to try and correct behaviors. No, all of us has moved their collection facilities to off reservation, specifically because they can use that loophole of tribal sovereignty doesn't exist outside of the reservation boundaries. So, nope, off reservation, there's no governance anymore. So they can do whatever they want. All right, well, I think we're on time. Thank you.